Hello, my name is Jan van Hauwermeiren. I am Business Development Manager at Ovizio. Thank you for watching this webinar. Today we will be talking about the viral load measurement and how this can be done in line with one of our instruments. Ovizio is well known for its capabilities of making 3D images or 3D holographic images instead of 2D holographic images. And from the 3D holographic images we gather from the cultures, we are able to analyze, identify, count, and assess the viability of the cells in your process. So next to our patented measurement principle to look at the viability of a cell, we have our differential digital holographic microscope, of course, that gives us images in a continuous manner as a live phase image capture. And from those things, we can calculate all the results and all the things that you are looking for. So what are the images that we can provide to you? So on the upper left, you see the intensity image. Intensity image is just like a bright field image, but because we are not using any stains, it's really hard to see something in that image. Then, of course, we have our phase image. The phase image tells us already a lot more on the cell and on the culture, but what's really important is the 3D holographic image. So the 3D holographic image, where you see in the bottom middle and the bottom left in a top view, that's where we extract really all the information, of most of the information. And on top of that, we are also able to refocus on the cells who are not completely in focus once the image is captured. So the refocusing capabilities you see playing in the small video on the top right. So how do we do all this? So how do we analyze the images? First step is, of course, acquiring the image and acquiring the hologram. Then we have a computation phase. We detect the light cones inside the cells. We refocus on all the objects that, of course, are out of focus. And then we extract what we call the fingerprint, so the holographic fingerprint. All this takes roughly around 30 seconds, so it doesn't take a lot of time. So the holographic fingerprint for cells basically is an algorithm that can differentiate different cells based on all the parameters that we are recording on a single cell level. So there are 70 parameters. Some have to do with the system as a whole. Some have to do with the morphology of the cells, the optical parameters, phase texture, or the intensity texture. Some are really well understood by everybody. Others have a better meaning for the algorithm and for example, optics experts. But that holographic fingerprint allows us to really identify cells, also differentiate between two different cell types, differentiate between living cells and dead cells, and so on. On our parameters, we use what's called a machine learning methodology to really build an algorithm that's fit for a specific cell type or for a specific purpose. So the first step in machine learning is that you create a learning set. The learning set can be created by any culture that you would like to investigate. So we start with 100% living cell and 100% dead cell, or 100% non-transfected cells and 100% transfected cells. The second step is that we feed the system with known mixtures from 100% to 0% in steps of 10. And then we basically have our finished algorithm, and we can start using it to predict our analysis and to predict the results in our culture. So the single cell analysis and do really the real-time measurements. How does it look like in real life? So what you see now is a screenshot of an ongoing analysis. So this is basically the result at the time point 56 hours in a bioprocess, where you can clearly see the viability and the viable cell density, and where in the middle you can see look at the cells and see the cells as they are moving through our flow cell. In green is indicated in this case the viable cells, in red the dead cells, and in yellow the aggregates. And then completely on the right, you can also see that we can make distribution graphs on any of the 70 parameters that we assess. In this case you see the distribution graph on the diameter. So you can also click on a cell and then you get the results, the individual results on all the parameters of this specific cell. And you can also make a scatter plot on any of 2 parameters that we are using to look or to analyze at a culture. All the results can be exported into an Excel file or a CSV file for further exploration. But in the end, what we really offer is continuous monitoring, of course, of your culture. So that means that you can instantly get the graph on the viability and the viable cell density and total cell density, and you can instantly see the results as well, the real numbers on the viability, viable cell density, or anything else that you would like to know about your culture. So the statistical relevance of the continuous monitoring is really high, as we take with our device 1,200 images per day, which is a lot more than a regular cell counter who only takes 50 images per day. And on top of that, we do not require any dilution. One other feature that we have is that we can incorporate our software into any OPC-ready software to create feedback loops, or via a web browser or a web interface to look at your culture from a distance. So you do not have to be in the lab during the weekend or at night to look at your culture. You can just log into the system and have a look if everything is okay. The system can also send out an alarm when, for example, viability is dropping or spiking, or when aggregates are dropping or spiking. All of that, of course, means that the device, the iLineF, can be used as a PAT tool and can be used to automate a whole process, the whole process of cell culture monitoring. The benefits of automation are well known. So there is, next to the reduced investment and the reduction of manual operations, of course, the increased control, the time gain and traceability of the results, and the increased reproducibility of your results. But today, maybe the most important is the case study that we finished a couple of weeks ago, the viral load measurement. So we set up an experiment with insect cells and a virus. We have repeated the experiment, in this case 3 times, where the cells got infected the first 2 times after 72 hours and the 3rd time after 80 hours. And the goal of the experiment was, can the holographic fingerprint be used to determine the viral load progression during the culture? So the basic results of all 3 experiments, when we are looking just at viable cell density or viability or the diameter, there we see that we are in line with other cell counters or cell monitoring devices. So there is the regular trend line when it comes to VCD, and there is the drop in viability at the end of the run and at the end of the process. So the same for experiment 2 and the same for experiment 3. So all 3 experiments were totally normal cultures, where the cell density went up and the viability in the end went down, and where there was a small spike in diameter growth towards the end of the culture. So what did we do? We used the machine learning methodology to look if we could find something that could determine the difference between an infected and a non-infected cell. So the cell images of the beginning of the run were used to create the virus-free training set. The cell images at the end of the run were used to determine the viral load fingerprint. And then we used a filter to get an optimal training set. And infected cells actually clearly show changes on the holographic image that cannot be detected by any other method. That's also what you see in the 2 small pictures, where there is clearly a difference between a non-infected cell and an infected cell. And even with the limited training set of those 3 experiments combined, we achieved a false positive and a false negative rate of under 10%, which is quite impressive, of course. So here you see screenshots of the viral load training set. So on the left, you see the images at the very early stage, where in this case, green indicates that the cells are not infected. and red dot indicates that the cells are infected with a virus. You can clearly see this on the right image. As this is a phase image or even a regular bright field image, there is not much of a difference to see between a transfected and a non-transfected cell with the naked eye. But if we go to the holographic images, there we can really see what you could call dimples on the surface of the holographic image that indicates this is an infected cell, a transfected cell. So at the experiment end. And this is what the results were when we set everything out on a timeline. So we have our parameters that were defined by the machine learning methodology. We looked at them during the experiment, and then we can see that the transfected cells, the percentage of transfected cells really went up, and it starts around 72 hours after the beginning of the experiment for experiment 1 and 2, and a little bit later for experiment 3. So the conclusion is that the experimental goal was met. So the iLineF technology and the holographic microscopy can be used to monitor viral load in real time during a bioreactor run. So it's an online real-time monitoring of not only the viable total cell density and the viability, but also of the viral load percentages. So this allows anybody to optimize their viral procedures, because it can be done for any virus produced in cells. So we are using a single-use disposable BioConnect that reduces the risk of contamination, which is consistent with MFG procedures. And it's a non-invasive, label-free system that never contacts or consumes any of the culture. So this is, I would say, new technology that can directly be implemented in any processes looking at the viral load in suspension cell cultures. So I would like to thank you to listen to this presentation and this webinar. We would love to continue the discussion on this experiment with you. If you would like to contact me, you can do so. My contact details will be on the next slide.